Hi, everybody. This is Tyler Shelpat, your guide to the wonderful world of antibiotics. The topic of today's lecture is the molecular mechanisms of antibiotic resistance. Antibiotic resistance is a really important topic. Antibiotics have widespread practical use in medicine and in treating bacterial infections. And because of this widespread use of antibiotics, bacteria have developed resistance mechanisms, or these resistance mechanisms have emerged as a way of combating the effects of antibiotics. So the topic of today's lecture is molecular mechanisms of antibiotic resistance. So we'll be exploring specifically what is happening at a molecular level that confers antibiotic resistance. The first question that must be answered is, what is an antibiotic? These are two different definitions of what antibiotics are. The first definition is that an antibiotic is an antimicrobial agent produced by microorganisms. One major question that must be addressed is, why do microbes produce antibiotics? This definition illustrates a key point about antibiotics. They are mostly natural products, save a few that are synthetically produced. Essentially, antibiotics are chemical compounds produced by microbes to kill other microbes around them, making them more competitive for nutrients and increasing their fitness. A second, more functional definition of what an antibiotic is and its function comes from Selman Waksman, a microbiologist who studied antibiotics. He suggests that antibiotics are agents that inhibit the growth and metabolic processes of other bacteria. This is the general way that antibiotics kill bacterial organisms. They inhibit essential processes in bacterial cells necessary for growth and cell functioning. A final point that must be addressed is the nature of antibiotic resistance. If bacteria produce such potent compounds that inhibit growth and cell functioning, how can they survive and not wind up killing themselves? The answer to this question is that antibiotic resistance must exist naturally to protect microorganisms that produce antibiotics from inhibiting their own cellular processes. Resistance is the result of genetic phenomena, such as mutations that prevent antibiotics from binding within cells in which they are produced. Other cells, by virtue of mutation in special systems, can also be resistant to antibiotics produced by other organisms. Because resistance is genetically based, it can be selected for by natural selection and be conferred from bacterium to bacterium through the process of sharing genetic material, otherwise known as horizontal gene transfer. Thus, resistance can be innate or acquired. Bacteria are able to grow and divide because of essential cellular processes such as protein synthesis, nucleic acid synthesis, cell wall production, and metabolic pathways that allow cells to harvest energy from the environment and convert it into a form they can use. Antibiotics function by binding to specific proteins that carry out these pathways and inhibiting their functioning, thereby inhibiting the pathway and preventing the cell from growing and dividing. On the left is a picture of a bacterium undergoing cell division. For cells to divide, they need to synthesize new proteins, build more cell wall and cell membrane, and harvest energy through metabolic pathways to drive these processes. The bacterium also needs to replicate its DNA so the daughter cells will each have a copy. DNA replication requires nucleic acid synthesis. So by inhibiting a process like nucleic acid synthesis through the use of an antibiotic, the cell will not be able to undergo cell division. This is one example of how antibiotics can inhibit cell growth and proliferation. It is also important to understand the role of genetic material in cellular functioning, because alterations in genetic material are much of the basis of resistance. Genes encode for enzymes and other proteins essential for cell growth and division. These enzymes carry out specific, essential processes that are inhibited by antibiotics. Antibiotics work by binding to these proteins, that's a key point, and inhibiting their functioning. Thus, the genetic material and what is encoded in this material provides proteins with specific structures and functions which are exploited by antibiotics that bind to these proteins with various efficacies. The main point is that the basis of resistance is genetic, which we will observe in the following slides. As we've learned, antibiotics work by inhibiting bacterial cell processes. One question we must ask is, why are antibiotics specific to bacteria? Why do they not kill eukaryotic cells as well? If antibiotics were generalized for both bacterial and eukaryotic cells, then taking an antibiotic for a bacterial infection would be disastrous because it would not only kill the bacteria, but it would cause serious detriment to the host as well. Why don't antibiotics harm eukaryotic cells? On the right is a table of bacterial cell processes and the targets of some antibiotics with an example of each. Antibiotics work in a few ways and are specific for bacterial cell processes. Antibiotics do not harm eukaryotic cells because they inhibit processes that eukaryotic cells don't have or that are carried out by enzymes and proteins that are distinct from bacterial cells. In other words, antibiotics may inhibit nucleic acid synthesis in bacteria but not eukaryotes because very different enzymes catalyze these reactions in each cell, and antibiotics, being microbial in origin, only have efficacy for the proteins that catalyze the bacterial reaction. Another example of this is protein synthesis inhibition. Some antibiotics function by binding to the ribosomal subunit and preventing protein translation. Because bacteria and eukaryotes contain different sized ribosomes with different structures, antibiotics can inhibit the ribosomes of bacteria but not eukaryotes. Antibiotics can also be specific for bacteria because they inhibit pathways that only bacteria use and that are not present in eukaryotic cells. For example, bacteria synthesize cell walls of peptidoglycan, which eukaryotic cells do not have. Antibiotics can inhibit this process, which, as stated, is absent in eukaryotes. Bacteria also utilize metabolic pathways not found in eukaryotes, so the proteins that the antibiotics bind are only found in bacteria and eukaryotic cells are unaffected. So, antibiotics are specific for bacteria and don't harm eukaryotic cells, which makes them practical for situations where a bacterium is infecting a eukaryotic host organism. The bacteria can be killed without damaging the host. Once again, there are only a few ways that antibiotics work. It is limited and not infinite. There are many antibiotics that fall into a few classes based on their function. This is a diagram of the mechanisms of antibiotic resistance that can exist in bacteria. Just as antibiotics elicit their effects in only a few ways, there are only a few ways that bacteria can be resistant. Bacteria can resist antibiotics simply by preventing their entry through a non-permeable barrier. If the antibiotic cannot get inside the cell, there is no way it can bind its target. Bacteria also contain genes that encode for efflux pumps. Basically, these pumps are located in the plasma membrane and remove antibiotics from the cell by pumping them back out into the ECM. Removing the antibiotic ensures that it cannot make contact with its target. Another mechanism of resistance is acquiring a resistance mutation that alters the structure and conformation of the antibiotic's target, which reduces binding efficacy and affinity and allows the enzyme to perform its function without being inhibited. Finally, bacteria can use enzymes that are already present in the cell or acquired genetically that degrade the antibiotic, thereby rendering it useless and preventing from binding its target. Basically, to be resistant, bacteria have mechanisms that help prevent the antibiotics from binding their targets. Let's examine these mechanisms in a little more depth. We know that bacteria can acquire mutations that alter the drug target and decrease the binding efficacy and affinity of the antibiotic. Suppose the red circle represents an antibiotic that binds to the yellow enzyme, whose structure is encoded by a genetic sequence on the left. By altering the genetic sequence on the left, the structure of the enzyme can be altered and the antibiotic no longer binds to it or doesn't bind to it as effectively. For the sake of simplicity, only one base pair alteration is shown, but in reality, altering the structure of an entire enzyme may require many mutations throughout the coding region. Antibiotics work because they can bind to proteins based on structure, so altering the structure changes binding. You may be asking, why does a mutation that leads to resistance not kill the bacteria with the mutation? What if the mutation affected the structure of the enzyme to the point where it could no longer function? This is a good question. Mutations in the coding region for the protein may be detrimental and the protein may not function as well. These mutations must occur in such a way that together they only slightly alter the protein to reduce the binding efficacy of the antibiotic while still maintaining enough protein function so the protein can do its job. In this diagram, alteration is drastic, but in nature, alterations would not likely alter the structure of the protein to such a degree. If the bacteria acquires a set of detrimental mutations that prevent the protein from functioning, their fitness may decrease significantly, whereas a bacterium that acquires a set of mutations that keep normal functioning of the protein while preventing the binding of the antibiotic will be selected for and passed on to daughter cells, conferring resistance down the lineage of that bacterium. Remember, mutations are totally random and can be detrimental or beneficial. Bacteria can also resist the effects of antibiotics by swamping the antibiotic. That is, producing so much of the target that even though the antibiotic can bind and inhibit some of the target, there is still enough unbound protein to carry out essential processes in the cell. Suppose the red gene in this coding sequence encodes the red circle, the enzyme, which is the target of the antibiotic in blue. If all the enzyme is bound by the antibiotic, function is inhibited. However, if the enzyme is overexpressed, as diagrammed below, then there is enough free enzyme to carry out the essential processes, even though the antibiotic is bound to some of the enzyme. The overexpression of an enzyme can occur through gene amplification, where the gene encoding for the enzyme is duplicated or amplified, thereby amplifying the amount of protein for which it encodes. Bacteria may also contain enzymes that over many generations have become specialized for binding antibiotics and degrading them, thereby inhibiting their functioning. An example of this is beta-lactamases, which degrade beta-lactam antibiotics, which inhibit cell wall biosynthesis. The structure of these two enzymes is presented on the left. Genes encode for the enzymes, which then bind antibiotics and degrade them, as shown on the right. It's as simple as that. Efflux pumps can prevent antibiotic functioning by leaving the antibiotic intact but removing it from the cell altogether so that it can't reach its intended target. On the left is a crystal structure of one efflux pump encoded by genes in the bacterium's genome. On the left, notice how this efflux pump spans from the cytosol to the extracellular matrix spanning the periplasm. It is composed of 3 subunits, two embedded in the membrane and one that links those two. On the right are more examples of these efflux pumps. Notice that they require ATP or an ion gradient to pump the antibiotic out of the cell. These pumps require energy. Now I'm going to talk about the major way resistance is conferred. We have learned that bacteria can acquire mutations that alter the binding target of the antibiotic, but bacteria can also acquire genes encoding for things like beta-lactamases and efflux pumps from other bacteria through horizontal gene transfer. For the sake of simplicity, we will only address the most basic way that horizontal gene transfer can occur. When bacteria die in lice, they release genetic material into the environment. On one of these chunks or segments of DNA may be a gene that confers antibiotic resistance. Another bacterium in the surrounding environment can pick up this segment of DNA and incorporate it into its genome. This is known as transformation. There are other ways horizontal gene transfer can occur, but they are not important right now. The key point to understand is that bacteria can acquire resistance from genetic material of other bacteria. Thus, populations can share DNA and can confer resistance throughout a population. So let's review. The basic mechanisms of resistance can be acquired through inheriting DNA or be innate through mutations that confer resistance. Antibiotics work in a few ways. Antibiotics inhibit essential processes through binding. Protein binding is most important. And antibiotics are specific for bacteria, which makes them especially effective in treating infections that are bacterial in nature in eukaryotic organisms. Just as antibiotics only work in a few ways, there are only a few ways to be resistant. These include the modification of the drug target, swamping, degrading, and removing the antibiotic from the cell. Thank you very much for listening to my presentation. I hope you enjoyed it, and I'm signing off. See you later.